All right, good morning. We'll get started with our omics panel. If y'all take a seat. All right, so in the interest of time, my name is Maggie Hunter. I'm with the U.S. Geological Survey, and I'm one of the co-leads for the Genetic Composition Working Group here in GEO BON. And it's my honor to introduce the Omic BON. They've been around for a while, but this is the first conference that we're able to introduce them. So they're the first thematic BON focused on collaborative efforts between national, regional, and global observing systems, also standards and best practices organizations, and data and sample management infrastructure for omics. And just to define omics, it includes all of our DNA and RNA sequences, proteins, metabolites, and other biomolecules. So today we'll talk about the opportunities for integrating these technologies, as well as the challenges that come along, and the need for standardization and interoperability across different observing systems. And so Raisa is going to lead us off with a short presentation on the overview of Omic BON.
Thank you very much, Maggie, and thank you everyone for being here. As Maggie mentioned, our biodiversity observation network is about omics, and the motivation behind that really is very similar to, I guess, what the other biodiversity observation networks had. That while we know that biodiversity is changing on a global scale, we don't currently have the means to monitor and assess that routinely and in a standardized way on a global level. And we have that same problem with omics. And so we want to be able to get to a point where we can ask the big questions at a global scale and get a better understanding of how everything is changing. And what is so special or unique about omics is that it doesn't only cross-cut all of the different ecosystems, but it also allows you to view diversity at all of the different levels. So from species diversity, genetic diversity, species diversity, up to the ecosystem. And that brings a lot of opportunity, but we also have to really make sure that we're navigating the space properly, because there are so many actors coming in, and now we're at a critical point in time where we can organize it, and we can bring all of those actors around one table to make sure that we're building something that will be sustainable in the future. Okay, so the development of Omic BON, just really quickly, this was kind of started off with the unification of two existing omics networks, and then we brought in a bunch of partners from long-term observatories, from data and sample infrastructures, as well as standards and best practices organizations, and global ocean observing programs. I'm sure a bunch or some of the logos seem you're familiar with, and then others maybe are a bit new because they're really based on what we're doing with omics. And all of that then led to Omic BON being created under GEO BON, so we now have that home. And most of you probably know how GEO BON is organized, but as Maggie already mentioned, we're a thematic BON, and we're the first thematic BON that's focused on an observational technique, really underscoring the importance or the capacity or, the importance of what we can do with omics. Okay, so what are we about? The overarching goal of Omic BON is really to build a sustainable and responsive, globally integrated omic observing system so that we can monitor biodiversity at the biomolecular level. And part of that is transitioning the fragmented observations that we oftentimes still have into a more coordinated contribution to a meta-observatory. More specifically, I'm just going to show some early priorities and long-term goals. So we have been in existence for 1 year, and the early priorities really are, for one, to set up an operational BON, but then we're really considering how important it is to have a data strategy and a strategy for tracking and indexing samples from the get-go, so that we already have that landscape kind of cleared and everyone knows what's expected and how best to contribute. And then long-term goals range from just having a forum so that we can continue to coordinate omics observation up to having the nimble response capacity and export of data products such as the essential variables, and a whole bunch in between. And now I'm just going to leave you with a very simplified picture or image of our landscape that we're in, where we have the observatory-grade activities, global observing programs, data and sample infrastructures, etc., and just show where the partners that we're currently working with are sitting. And we're really lucky to have some of those partners on the stage today. And I think with that, I'm actually going to head over to Maggie again, and everyone can talk a bit about how they fit within biomolecular observing and Omic BON. Thank you.
All right, thank you. Okay, so we will spend just 2 or 3 minutes each introducing the panel members, and also please, like Grace has said, describe your role in biomolecular observations and how you fit into the Omic BON landscape. So Chris, we'll go ahead and start with you.
Is this on? Yeah, good. Hey everybody, my name's Chris Meyer. I'm from the Smithsonian's Natural History Museum, and I've been involved with the Genomic Observatories model at the beginning, did a lot of sequence-based work, and also represent Marine Geo, run a bunch of networks, and just really quickly, a few quick lessons, overarching things about building systems. It's an incredible time because we're catching this new technology, and so harnessing and aligning and coordinating that early is going to be critical, and I think that's what Omic BON intends to do, is standards are critical, a data management plan, and keeping things simple and returning the information back as quickly as possible to those in need. And those are the big challenges that we've seen in building this. And it's really going to be really important for Omic BON to provide that. People just want to know what to do too, just like we're like a bat phone or a clearinghouse for that kind of information. It's the critical role. So sign up and get involved, and because it's moving fast, and we want to kind of keep it aligned so we can make the sum greater than the parts.
Yeah, and I'll just mention you can sign up by going to the GEO BON, Omic BON website and registering to get on the listservs. All right, we'll move on to Katerina. Thank you.
Okay, hi. My name is Katrina Exter. I'm from the Flanders Marine Institute in Belgium. And I'm here as part of EMBRC, which is a European marine biology research infrastructure which covers many different countries within Europe. And EMBRC, a few years ago, started a project called GEO BON to do regular, harmonized, standardized sampling in the marine environment around Europe. We have 16 sites. We collect every 2 months. We have standardized protocols, methodologies, and we're starting to produce the sequences and starting to analyze those sequences. Now, the main aim of GEO BON has always been to have regular, long-term, standardized sampling, storing, processing, sequencing, and data management. So we put a lot of effort into making sure that our data and our tools are fair and open access. Our protocols are published, so that's the physical, the wet protocols and the dry protocols, the digital protocols, if you like. So anybody could see them and could choose to adopt them, and also benefit from our data management experience and our data management systems that we're also building. So as well as collecting samples and creating data, and therefore science, we want to be an example to others, to encourage the adoption of doing fair and open science and following standardized methodologies and processes and the data management for bonds and for any kind of long-term sampling activities. And we want to be a resource for others, particularly within Europe, to learn from and to use. We want to implement and to push and to encourage the development of community standards in the field of eDNA, partly because we need this in order for us to do our work, so that our data are interoperable with other data, or more, so that other data are interoperable with our data. But also because for the data and the science from these long-term monitorings and bonds, for them to get out there sort of rapidly and effectively, it's necessary that, that you do these things in a standardized, harmonized way.
It's on. I'm Sujeevan Ratnasingham. I'm from the Center for Biodiversity Genomics, and I oversee the BOLD and mBrave platforms that Paul Hebert just talked about at this morning's session. I'm a recent addition to this group, the Omic BON, where we provide perhaps a unique perspective, is that we've got infrastructure that's mature. We've been working with the community on an arguably a narrow section of the landscape of data and practice. We've been looking at systems that support the generation, validation, and publication of DNA barcode records. And so that focus has given us a unique trajectory. We started off with by establishing data standards very early on in 2005, and we brought in partners to ensure that those standards were not just guidelines, but were also enforced by having organizations like GenBank deal with the tail end of the data lifecycle, where data would be published, ensuring that those standards were met. We also focused very heavily on supporting data generators by working very closely with them and providing analytical functionality, data storage functionality. And on top of that, we also added support services and annotation and curation support. And that became really critical because our focus was not simply collecting data, but producing the reference data that would support the generation and annotation of all the other data that people were collecting. So my hope is that that unique role of focusing on reference data is something that we can provide to this group.
Yes, important point. Lori.
Are we on? Okay, sorry about that. Hi, I'm Laurie Phillips. I'm a research scientist with Agriculture and Agri-Food Canada. I am a pure bench scientist at this point with a heavy interest in using, obviously, molecular tools within my discipline. And I think within, having listened to the talk by Paul this morning, within microbial ecology, we were obviously early adopters of molecular tools and using sequencing approaches to evaluate the systems that we're working on. They're the best tools for us. Within Agriculture and Agri-Food Canada, we are interested in establishing what we're calling agricultural terrestrial genomic observatories. AAFC is involved in Omic BON for this purpose primarily, although we certainly have interests in all of the other fields. AAFC is a custodian of over 20 long-term agricultural research sites across the country. Some of these are older than a century. The one at my station was established in 1959. All of these agricultural sites will have complementary non-agricultural sites associated with them and or other best management practices associated with them. Within Canada, we're seeing climate change advancing quite rapidly as we go from the south to the north. For us, managing and mapping how soil biodiversity changes as climate change progresses is critical. And AFC, we believe, can play a really important role with these genomic observatories based in protected agricultural lands that are managed at a much higher level than universities can manage. No offense to universities, sorry. But long-term research is obviously where government organizations excel, and so we're interested in having these genomic observatories, again, embedded in a larger scale. So our interest in Omic BONs are multiple. First, increasing awareness of the availability of these systems within Canada. And I'm only mentioning one level of observatory that we have within AAFC. There's certainly many others. But so increasing awareness of those observatories, increasing usage of those observatories to answer different questions. Most of these sites also have archive samples, either dried archive samples or frozen archive samples, dating back in cases, over a century, that can be used to investigate questions of change as climates shift. And I think in agricultural settings, when we're looking at beneficial microbiota within soils and pathogenic microbiota in soils, being able to monitor temporal changes in these organisms as the systems shift is critically important for food security across the country. So again, using Omic BON as a tool to help support interoperability of data across these sites and with global sites who are monitoring the same thing, leveraging Omic BON to help us articulate the value metrics associated with maintaining these sites is critical. As you can imagine, maintaining long-term sites is not a trivial matter. It's very expensive, it's very time-consuming, and we constantly, as many of you probably have to justify the expense of maintaining these sites to our managers. And Omic BON, I believe, can play a critical role in showcasing the value that these sites provide in answering questions of sustainability in the long term. And so I think those sort of two main points, the interoperability of data. We try our best within Canada, but we're a very large country, and there's a lot of siloed projects going on these different systems. So having protocols that are relatively standardized, data that's interoperable, metrics for storing that data and metadata in a meaningful way, and adding those value-added metrics so that we can continue to support these sites to answer questions about how our systems are evolving as our climate shifts are all critical.
Yes, hello everyone. I'm Tim Hirsch, Deputy Director of GBIF, the Global Biodiversity Information Facility. As many of you will know, GBIF is a global infrastructure essentially made up of government representatives and nodes throughout the world. And our overall aim is to bring together all available data that helps to describe essentially the distribution of life on Earth. Now, we've realized for some time that if we're really going to fulfill that mission, we need to go beyond many of the classic traditional ways in which that information is brought together. For example, the digitization of specimen collections and the bringing together of observations of many different kinds. And in order to keep up with the patterns of how data is brought together, we need to find ways of properly and efficiently combining all those more classically derived data sources with this increasingly sophisticated and exciting world of sampling at the molecular level. So for some time we have been collaborating with, for example, with BOLD for many years, also with the INSDC, mirrors in the GenBank and the European Nucleotide Archive, also groups such as the Unite Fungal database, and more recently the Global Bacteria Taxonomy database, to look at ways in which we can most efficiently interpret and ingest and bring in data that is coming through from these different sources. And most recently, we are currently, for example, piloting a new tool whereby data coming directly through eDNA samples through ASV data can be directly mapped and interpreted to the kind of records that can then be served into GBIF without the sharer of that data having to do a lot of mapping and squeezing of that data to get it into our standards. So increasingly we're working with this world really to try and create the most efficient crosswalks and tools to be able to incorporate all of the data and the massively increasing volumes of data that's going to come from these sources, and to be able to make it truly interoperable with all of the other evidence and information that we have from the more conventional sources into GBIF. It's a rapidly moving world. There are exciting things coming up in some of the tools that we're planning to develop, but perhaps I should leave it at that and just say that I think in respect to Omic BON, I think we would see ourselves as a support service to help to ensure that this community, this observing community, is able to make use of our global infrastructure. Thank you.
Great, Raisa.
Thank you. That was very interesting to already hear from all the panelists. Maybe so I'm wearing multiple hats at this panel discussion, and picking up on what Tim said about the data standards, I think that's where I'm going to start. So I'm representing both TEDWIG, the biodiversity data standard historically, and also the GSC, which has developed its own standard that was largely parallel or developed in parallel with the Darwin core standard from TEDWIG, but very much focused on genomics, metadata, and data. And what I see here, maybe even like exactly between Laurie with you're in the lab, you're getting the data, and then you have to push that out somewhere, going over to GBIF, where you want to take all of that data in, but you need a way of efficiently doing that. And I think that's exactly where those metadata standards are coming in. And where also some of the work that we have been doing on mapping and harmonizing these two previously somewhat competing data standards even, one being used by GBIF and the other one being used by the INSDC. Really have that chance of already bringing those communities together and going with the time, because now that we can feasibly use omics observation to assess biodiversity changes, that needs to be reflected in the data standard space as well. Yeah, so I think that's on those two. And then Chris Meyer and I are both also part of the Ocean Biomolecular Observing Network, which is a program under the UN Ocean Decade, which many of you may have also heard about before. And I thought, before we go into the panel discussion, I'm just going to address one of the questions that always comes up as to, okay, so are you the same? How do you relate to each other? And kind of talk about how OBON sits in that landscape around Omic BON. And what we have found really is that OBON is, in a way, a marine component with, of course, standing on its own and doing incredibly important work on its own. But in the context of working with Omic BON, we can leverage all of the momentum that is in the ocean space with the UN Ocean Decade and the work that is being done within OBON to already lift up what we're doing in the marine space. Because, of course, Omic BON is cross-cutting ecosystems, so including marine, freshwater, soil, as well as atmospheric. And I think for now I'm going to leave it at that and hand back to Maggie.
All right, thank you. The breadth of the work and the focus is amazing. We really, there was a number of mentions of what Omic BON can do, and I think the different places of sustainability and helping to justify some of this work. Are there any other sort of gaps or opportunities that you see Omic BON coming in and helping in your work that weren't mentioned so far?
I'm just going to pick that up again and then hand over to Katrina, I think. One thing that also with this panel I think is really critical is how Omic BON can serve both as a purpose in a way of saying, okay, so there are people who are already doing biodiversity observations and would like to get into the omics space and would like to leverage that incredibly powerful tool set to assess biodiversity at all the different levels. And I feel like that's one of the critical roles that Omic BON can play, is to kind of reach out a hand and say, okay, we have the partners who are doing all of this on this stage and also in their offices at home at the moment. And we can find ways to make it easier for anyone who wants to get into omics observation to do that. So I think that's one of the sides. And then, of course, the other one is, even if you don't want to produce the data or do the observation yourself, Omic BON can play a role in then producing those data products and offer the information that we gain from omics observation to a breadth of stakeholders.
Yeah, even if you're not aware that it might be useful in your work, this is a place to come and find that collaboration or interoperability. Yeah, great. And then did you have something to say?
Yeah, I was going to echo, in fact, a lot of what we've all said. and particularly for EMBRC, what is important to us and what we can also offer is standards. From the wet, for the marine, to the dry, to the digital. Standards for the sampling, standards for sample processing, for storing, standards for the sequencing, and then standards for how you manage the data once you have sequences. And I mean technical standards, so things like data formats, metadata formats, but also content standards, so the content of the data, making them machine accessible, machine readable, the content of the metadata, using vocabularies, semantics, and then also the scientific content, so that that you give enough provenance along with data so that someone can understand how they can use your data for their own particular research, how they can combine the results of one BON with the results of another BON, but also agreeing on standards for EOVs and EBVs so that data can be compared. In these days of climate collapse, it's really, really important, especially for the ocean, because there is one ocean, covers the entire globe. It's really important that we can do whole Earth studies and that we can combine instantly the data coming out of one BON with the data coming out from another BON, that we know what we're looking at and that we can instantly combine. And we have the power to do that now. We can do it. We have the technology. It's the human side of the equation that's missing. And I think this is something that we can do here with Omic BON. We can encourage people to adopt these standards, but also help them. Perhaps find a way of developing tools that can help individual scientists and help universities and labs to do all of this standardization, especially at the data level.
All right, perfect. And with just 5 minutes left, we've got some amazing sessions coming up on eDNA and biomolecular observations the next 2 days. So what are some of the advancements you're looking forward to in these talks? Are there any kind of metrics or interoperable advancements that will allow for these data to be used now to inform conservation and management efforts, and then also into the future where these data could be used for things we don't even know about yet? No.
Turned it off. Oh my gosh. I just want to follow up on that a little bit and lead into it. I work at collections. We're the Natural History Museum. We store these collections. These are time capsules. Like, we can't go back in time. And so the importance of thinking about the future use of these, the technology is going to change. So making sure we lock down the context. We need and I'm going to be listening to all the talks about, usually what's nice about collections, for the environmental collections, is they get used for a purpose usually, right? A lot of people don't just collect environmental samples and let them sit around. People dredge stuff and let them sit in museums and they never look at them, but these usually get looked at. But they can get looked at again, right? And so thinking about fair and care principles of the material samples as well, baked in up front, is really important to think about in how we negotiate permits, work with land folks, work across the spectrum here, and we need a strategy for that, again, because we know we don't want to have lock-in, we want to allow innovation and development, and so being able to see what's available to get this longitudinal perspective on change on the planet is going to be really important. So coming from a collections-based institution, that's a perspective I see as really critical, and it's a pinch point also that we, as holding institutions, can make decisions about. We'll take that sample if it has this metadata. So figuring out what are the critical metadata, the minimum metadata that are needed for samples. This is one of the spots, and I think, Tim, your spot where you take the data is another pinch point. So identifying those spots where we can kind of enforce harmonization. Sujim, you did it with BOLD. I mean, you have that experience. So I think those are really important spots right now as we move forward. Again, we can't go back in time and resample. And the real cost is the sampling. The time, that's an incompressible cost. The sequencing is just going to get cheaper and cheaper, and we'll be able to interrogate it with more and more tools. So that's...
Yeah, and access to the samples, being able to search for samples that are out there, and democratization of the ability to use those samples. Yeah, Tim.
maybe just in terms of kind of some of the exciting possibilities I think this all opens up, something which I think has been pretty amazing to see in a relatively short space of time is that because we are now able to map this data coming in from DNA samples with reference databases such as the fungal database that I mentioned, already what we can do from that is we can actually start to see geographic distributions of things that don't have a scientific name. They simply have a reference, something like a DOI, as an operational taxonomic unit. And even just in the start of some of the fungal DNA samples that we brought in and mapped against this database, you can see, well, hang on, this thing which has one alphanumeric code appears in that part of Denmark, and that other thing which has another alphanumeric code but maps to the same species occurs somewhere in Russia. And so you can already start to see that distributional information and perhaps some of the conclusions on that as, in terms of the functional biodiversity that you can see within species. You can actually start to map that ahead of species identification. I think that's got to be a really exciting area for the future. And just going back to Chris's point as well, just pointing to one area where we have plans now in moving on from the tools that we brought in. I think one of the things we're really keen to do is to make sure that we can always keep the latest interpretation and the taxonomies that are coming through with more and more eDNA being deposited, and to make sure that we can continually reassign those identifications from the samples that are deposited within GBIF with proper tracking through persistence identifiers to make sure that you have, if you like, version control of those different taxonomies. I think it's something that we know that there's a big demand for, and it's certainly something that we're intending to implement.
That's great. That capacity is great. All right, just 2 minutes maybe for each of you if you have final thoughts, and then we'll wrap up for the next panel.
Yeah, I think looking forward for myself, what I'm looking forward to seeing is how other people have managed integration of different projects and programs within this field. I've mentioned that we have the genomic observatories, if you will, in the long-term sites in AFC, but we also have a lot of other projects that are a little bit more siloed. My colleague Frank Stefani, for example, just finished an 1100 sampling regime across Ontario and Quebec of soil samples to try to establish baselines of soil biodiversity in both agricultural and connected forested sites. So we're looking to see what other people have done to try to integrate this type of data, how to move that data away from silos on desktops. We're working our best to try and develop computational methods and platforms to store this data in an outward-facing or a public-facing way, but there's lots of challenges associated with that. So obviously, Omic BON can help us with that, both storing the physical and the computational data, but more importantly, looking to see, again, with the next sessions, how other people have actually managed the same challenges in different disciplines. Because with soil biodiversity, there's a lot of work going on, but I would argue that it is still fairly compartmentalized, and certainly within Canada, we need to move away from that in order to answer bigger questions of the data. And there's a lot of information, as we all know, contained within that data, and how do we get it into a place and into a format and remove those institutional barriers such that those larger scale questions can be asked beyond the original questions that these samples were collected for.
Yeah, and hopefully Omic BON can help with that. That's a great thing. Okay, and yeah, our last comment.
Yeah, thank you. Well, since it's the last comment, I'll see if I can tie in a couple of the other comments that were made. And this is just from a perspective of having worked with the data generation community for a number of years. A key focus for us, and hopefully for the Omics BON, is on the reduction of friction. We know the endpoint that we want to achieve, which is lots of occurrence data points that are molecular-based, that's usable and meaningful to address various questions that we have. But trying to go after that endpoint at the outset can lead to increases the chance of failure. So by focusing on reducing friction, so what do I mean by that? is really tying into what Andy said at the outset of this meeting, that there are 3 levels of engagement. There's the community level, there's the national level, and the global level. And each one of those levels has different needs. Our strategy was start at the community level with reducing friction. And so if you break that down, they're really, on the standards front, there are 2 different types of standards activities. One is for minimum data. We need to keep lowering the floor on the types of data that we collect. And I think this group is well positioned to start to raise that bar. And the other is on interoperability. And these are 2 different things. And the interoperability should be both across our discipline in terms of data generation and data storage, but also broadly across like this community of Earth observation, making sure that our data points can match to the terms and ontologies that are used by the broader ecology community or modeling community. So I think by focusing on the friction points that people are experiencing now, we can have a great deal of impact very early on. One other area that I just want to touch on with regards to friction is onboarding. We want to set up a big tent where everyone can come in. Our landscape has gotten a lot more complicated. Technologies change very quickly. Data models change very quickly. A year goes by and you've got something new. And I know that the research community is continuously playing catch up. So I think there are a few things that we can do. One is to build onboarding tools, standard data sets, standard workflows, templates that people can start using. That will get more and more people into our group so that they can start contributing data. And then we can start bringing that community to the larger goal of global Earth observation and how we can match data up.
All right, thank you so much. Okay, we started a little bit late, so that was a quick session, but we do need to move on to the next panel. So I want to thank all of our panelists. You guys are all doing amazing work. We're really happy to have you on. And if anyone wants to sign up, please go to the GEO BON, Omic BON page, and you can sign up. Also, there is a Giga Science paper that was led by Raisa Myers, so please also check that out. That's been published here recently. And do you have any final thoughts that we want to cover? Okay. All right, thank you guys so much. We appreciate it.
Yeah, just because of the opportunity, I just wanted to really bring home that point that you also already mentioned about within GEO BON, we really have that opportunity to address what is a socio-technical challenge, where we now, with GEO BON, we can bring the people together, and then in the background, or also on the forefront, we make sure that the technical parts of that and the data flows and all of it also fit into each other. And we look forward to working with many of you on that. Thank you.